Welcome back. This is the beginning of the second video focused on the TCA cycle. We'll be beginning with talking about reaction 3. So reaction 3 is catalyzed by the enzyme isocitrate dehydrogenase. And this enzyme converts isocitrate into alpha-ketoglutarate by going through an oxalosuccinate intermediate. You'll also notice that this enzyme is a dehydrogenase, and as you remember, all dehydrogenases catalyze hydride transfers. Therefore, this reaction also produces a molecule of NADH, and as a part of this reaction, a molecule of CO2 is released. So here we're actually going from a 6-carbon isocitrate molecule to a 5-carbon alpha-ketoglutarate molecule. And because this reaction releases a molecule of CO2 and a molecule of NADH, you'll also notice that this is our second example of an oxidative decarboxylation. So the first example was catalyzed by pyruvate dehydrogenase, and the second one is catalyzed by isocitrate dehydrogenase. So this mechanism is going to require manganese or magnesium, which that should already tell you that this mechanism involves metal ion catalysis. So let's begin by taking a look at the mechanism. So if we look at the mechanism here we see isocitrate. So the first thing that happens as a part of the mechanism is that one proton is extracted from the hydroxyl group here, and then a hydride is extracted from this carbon as well. So the hydride takes its electrons, and these electrons are donated to NAD to form NADH. So now we've reduced NAD to NADH by oxidizing isocitrate. So because we took this proton here, these electrons will come in to form a carbonyl as a part of this carbon. So here you can see that now we have a ketone. So in the next step, the electrons from this carboxyl group are going to come in to form a double bond, which breaks the bond between these two carbons and releases a molecule of CO2. When this bond is broken, that pushes these electrons to form a double bond between these two carbons, which pushes the electrons from the carbonyl out onto the oxygen. So now we have two negatively charged oxygens that are really close to one another, which, as you can imagine, would be very unstable. However, this is stabilized by the metal ion. So the role of the metal ion in catalysis here is to stabilize the negative charge. So in the next part of the mechanism, the electrons from the oxygen are going to come back in to reform the carbonyl. This pushes the electrons back out onto this carbon, so temporarily this would form a carbene ion. However, this easily picks up a proton from solution, and now we have our product, alpha-ketoglutarate. The fourth reaction in the TCA cycle is catalyzed by the enzyme alpha-ketoglutarate dehydrogenase. So this is a dehydrogenase, so once again, that should remind you that a hydride is going to be transferred as a part of this mechanism. And in fact, that is the case. So here you can see that we reduce NAD to form NADH as a part of this mechanism. And in addition, we also release a molecule of CO2, of carbon dioxide. So we're going from the 5-carbon alpha-ketoglutarate to the 4-carbon succinyl-CoA molecule. So also as a part of this mechanism, the cofactor CoA is added to our 4-carbon molecule. So this mechanism should seem very similar to the pyruvate dehydrogenase complex reaction, and in fact it is. So like the pyruvate dehydrogenase complex, alpha-ketoglutarate dehydrogenase is also comprised of 3 enzymes. So the alpha-ketoglutarate dehydrogenase, dihydrolipoyl transsuccinylase, and dihydrolipoyl dehydrogenase. And in fact, this mechanism works almost identically to what you've already learned for the pyruvate dehydrogenase complex with the same 5 cofactors that are a part of the mechanism. So for any questions about this mechanism, just refer back to your notes about the pyruvate dehydrogenase complex. And now we'll move on to reaction 5. Reaction 5 is catalyzed by the enzyme succinyl-CoA synthetase. And the reaction that's catalyzed here is we're going to convert succinyl-CoA, we're going to release that molecule of CoA to form succinate. And as a part of this reaction, the energy from CoA hydrolysis is captured by adding an inorganic phosphate to GDP to form GTP. It's very important to note that this GTP can easily be converted into ATP without any major exchange of energy. So essentially, this makes a molecule of ATP in the end. So you'll note here that hydrolysis of succinyl-CoA has a delta G of approximately negative 32 kilojoules per mole, while we already know that ATP hydrolysis is also around negative 30 kilojoules per mole. So this is enough energy to supply to make a molecule of GTP and then a molecule of ATP. So in thinking about the mechanism, there were several lines of experimental evidence that helped scientists to determine what the mechanism of this reaction actually were. So first, the spinach enzyme, which actually uses ATP instead of GTP, showing that this reaction is highly exchangeable. This reaction transfers a phosphate from one ATP to another. So this suggests that there has to be some kind of phosphoenzyme intermediate where the enzyme can capture that phosphate and then transfer it to another molecule. Also consistent with the idea that there's a phosphoenzyme intermediate, it was actually found that a phosphate was attached to a histidine in the active site. So this should sound very familiar to you, as you've already seen a mechanism that has a histidine attached to the phosphate in the active site. So then finally, if we force this reaction to go in the opposite direction, so to go from succinate to succinyl-CoA, using labeled succinate as a substrate, so we use heavy oxygen in this succinate as a substrate, we can transfer this heavy oxygen to a phosphate. So this suggests that because we can transfer this heavy oxygen from succinate to the substrate, that succinyl phosphate is probably some kind of intermediate. So now let's take a look at the actual mechanism of this enzyme. So first, in the first stage of the reaction, what we do is replace CoA on succinyl-CoA with an inorganic phosphate. So as you remember, hydrolysis of CoA releases about negative 32 kilojoules per mole of energy. Therefore, this provides sufficient energy to add an inorganic phosphate to succinate. So now we have succinyl phosphate and we've released this CoA coenzyme. Second, we're going to transfer this phosphate from succinyl phosphate to a histidine within the active site of the enzyme. So here you can see that the histidine within the enzyme acts as a nucleophile to attack the electrophilic phosphorus within the phosphate. This breaks the bond between the phosphorus and succinate, so between the phosphorus and the oxygen, forcing the electrons onto the oxygen. You'll note that this is leaving the oxygen associated, that was once part of the phosphate, it's leaving it associated with succinate, which explains why that oxygen label could be transferred. So now we have a we have a phosphate attached to a histidine in the active site, and we've released our second product, succinate. So then finally, we're going to transfer this phosphate from the histidine in the active site to GDP. So here, the oxygen in the beta phosphate of GDP is going to act as a nucleophile to once again attack that electrophilic phosphorus. This breaks the bond between the histidine in the active site, forcing the electrons back onto the nitrogen. And now we have our guanosine triphosphate molecule. So now we've produced our last product, GTP. And that's the end of that mechanism. So we've now gone through 5 reactions of the TCA cycle. So in total, if we include the pyruvate dehydrogenase complex reaction, we have now produced 1, 2, 3 molecules of CO2 per pyruvate, which would be equivalent to 6 molecules of CO2 per glucose. We've also produced 1, 2, 3 molecules of NADH per pyruvate, which would be equivalent to 6 molecules of NADH per glucose. And we've also produced a molecule of GTP, which can easily be converted to ATP. So we've produced 2 ATP per glucose, or 1 ATP per pyruvate. So that'll be the end of this video, and in the next video we'll focus on these last 3 reactions, and we'll also talk about how the TCA cycle is regulated.